All right, I put on the Moodle site a set of kind of questions that might be asked on the exam on Friday, so you can kind of see how it's structured. You'll see that it's a combination of multiple choice questions and short answer questions, and short answer questions are basically asking you to give me information that you've been given. It's not asking you to think deeply about that information. It's not asking you to synthesize information, analyze information, et cetera, et cetera. So it should be a relatively straightforward test. Somebody emailed me asking if stuff from lab is going to be on the test. Stuff from lab is not going to be on the test. You essentially get tested on lab stuff when you write a lab report. So it's just lecture. It'll cover whatever we get through today. My hope is that we'll get through kind of how we go about testing hypotheses and asking questions in behavioral ecology. That's what is on the list. Great, so sorry.
Where on the list did you post those questions?
It's up at the top where all the course policies and everything are. And the part that doesn't ever change. It's there, correct? Well, I could have sworn I posted it. Well, that's weird. Maybe I didn't. I have it up on my computer. Yeah, I thought I had. I wonder why I didn't do it. Anyway, I'll get that up as soon as we leave. I had to find it on my hard drive. But anyway. Wait, or did I send it to you in an email?
No, that was a YouTube playlist, like you said that you put it on there.
Okay, all right, I'm not sure what happened there. Anyway. There it is, right there. I don't know why it's not on the Google side. I'll just go ahead and add it right now. That's easy enough to do. This is from a couple of years ago, or a couple of iterations ago, actually. So there's a question on here about blennies, which is this type of fish that lives in Southeast Asia that we read a paper about in previous semesters that we don't read a paper about any longer. You can ignore that blenny comment, that blenny question, but it's the kind of question that you might expect me to ask you about the Joseph and Pinchot paper or something like that. So, not all of the questions are going to be relevant to what you're doing, but I want you to kind of see the tone and tenor of the questions that are going to be on there. Oh, and there it is now. So it's in that section at the top. So if you go find that. All right. Questions from last time. All right, so we're going through the multivariate version of the breeder's equation, where essentially the evolutionary response to selection, in this case of reversals in these garter snakes, is due to the additive genetic variance of reversals being acted on by direct selection acting directly on reversals, plus the selection acting on stripedness affecting reversals through the genetic covariance between reversals and stripe. Same thing happens for stripe. You have the evolutionary response to selection is due to the genetic covariance between stripe and reversals multiplied times the selection on reversals and the additive genetic variance of stripe as it's being acted on by selection on the stripe. And so these are the actual numbers that are from Brody's paper. The additive genetic variance of reversals is lower than the additive genetic variance of stripe, so stripe has more genetic variation associated with it. And in this case, we're going to ignore the genetic covariances and assume that these two traits are not correlated with one at all. And so the way you get the evolutionary change is you multiply the strength of selection times the additive genetic variance, the strength of selection times the additive genetic variance. These are the two strengths of selection. Selection is much higher on stripedness than it is on reversals, and you get this as your outcome. One way of thinking about this is to superimpose this information on the adaptive landscape, where if we have reversals, few reversals to many reversals, and stripedness from checkered down here to striped up here, striped snakes with few reversals have high fitness. Checkered snakes that reverse themselves often have high fitness. The other combinations of color pattern and behavior have low fitness, and so you get this saddle-shaped fitness landscape, which is done here. It's just that we've taken that thing and compressed it down so that it looks like a topographic map rather than a three-dimensional landscape like this. And so if you start here, direct selection for reversals moves you a particular direction. It moves you towards more reversals. And direct selection on stripe moves you towards being stripier. And in this case, it takes you out of this low fitness area here up onto the plateau that's between this peak and that peak. Stripedness changes more because selection on it is stronger, and it is also more heritable. It has a higher level of additive genetic variance. Reversals moves less far because it has lower levels of selection and the additive genetic variance is lower. And you end up here on this plateau, you've increased in fitness on average as a population. And the evolutionary response to selection is the movement from where you began to where you end up in one generation. But what Brody found is that the genetic correlation between these two traits is very strong and negative. So as stripedness increases, reversals goes down. So there's this negative genetic covariance between these two traits within individuals. Striped individuals tend to go in a straight line. Checkered individuals tend to reverse themselves a lot. So when you add this genetic covariance in there, it tends to alter the outcome of selection because you now have this selection acting on reversals and you have this selection acting on negative genetic variance, which means that this moves reversals. Selection on stripe moves reversals much more so than selection on reversals moves reversals, and it moves it in a negative direction. So if we go back to this example here, we might see directional selection moving reversals one direction, moving stripe the other direction, but because of the negative genetic correlation, reversals is moved backwards because of the negative genetic covariance, and the stripe is moved backwards because of selection on reversals. The effect of this, once again, the effect of stripe acting on reversals is much greater because the selection on reversals is the selection on stripe is very strong and the genetic covariance is very large. The movement on stripedness is low because even though the genetic covariance between stripe and reversals is high, the selection on reversals is really low. And so what you end up with is you end up someplace else. You've actually, in this case, the way I've drawn it, you're a little lower in fitness than you were before, had you just been subject to the regular directional selection acting directly on traits, rather than correlational selection, where one trait affects the evolution of another trait. Logan, you have a question, it looks like.
Yeah, why did, when you're adding in this, the covariance, why are you moving from, not the original point, but from that point on the plateau?
Because what you have to do is this is just selection on reversals acting on the genetic variance of reversals. So this is a direct selection, so that selection acting on that trait. This is selection on stripe acting on stripe. So this
Oh, I
is,
see.
basically this section of the matrix algebra and this section of the matrix algebra. When we made the genetic covariance 0, that's all the evolution that went on.
Okay.
But this isn't the only thing that affects the evolution of reversals. Evolution of reversals is also determined by how much selection is acting on stripedness and the genetic covariance of that stripedness. So reversals is controlled by direct selection acting on reversals and correlated selection acting on the other trait as it's mediated through the genetic covariance. And because this genetic covariance is negative, it actually undoes what some of the direct selection actually accomplishes. So selection on stripe is strong, we saw, and because the genetic covariance is large, that ends up in a big change in reversals because of selection on stripe. The genetic covariance between stripe and reversals is big, but selection on reversals is relatively weak, and as a result, stripedness ends up changing very little because of this correlational selection. And now, the evolutionary response to selection isn't to go from here to here, it's to go from here to here, because you have direct selection and you have correlated selection. So here's the take-home point of all of this. When we start thinking about multiple traits, negative correlations can either cancel out or slow down evolution, reverse the direction of selection in some cases. Positive correlations can do just the opposite. If two traits are positively correlated, then selection on one trait is going to increase the other trait, and both traits will proceed at a faster clip in terms of their evolutionary change. But in general, the direction and rate of evolution is determined by the shape of the adaptive landscape that they occur on and the underlying genetic architecture of the traits that are object of selection. If traits are not correlated, then they're free to evolve separately from one another. But as soon as they're correlated, you can have one trait constraining the evolution of another trait because of these genetic correlation or covariances. So when I think about constraint, this is something that we come in evolution ecology. A lot of times you can think about constraint colloquially. For every advantage, there is a disadvantage. You can't have your cake and eat it too. Superman, perfect example of constraint. He can do all these cool things. Unfortunately, he has kryptonite, which is his Achilles heel. You can't go to the Chiefs parade and also be in animal behavior class at the same time. It's just impossible to do those things. You've allocated your time in one way versus another way, so you also represent constraints of different types. But when we think of constraint within evolution, we think about other things, not going to the Chiefs parade. There are morphological constraints. A classic example of this is the Carn and Penrose data from 1951. These are data on the birth weight of babies and the probability of survival of women in the early 1950s. And as you can see, if you're prematurely born at a low birth weight, your odds of surviving are relatively low. There's this plateau of kind of optimal birth weight where everybody does well. And then as you start to get too large, your probability of survival goes down because now you're too large to fit through the birth canal. You are constrained, women are constrained in how big of a baby they can pass because they have a pelvic girdle that's made out of bone. And if your baby is too large, it doesn't pass through that birth canal very well. And as a result, survival goes down. And if you're too small because you're born prematurely, you haven't fully developed, and that has lots of negative consequences. This is 1951 data, and this represents at this end of the spectrum, this is a morphological constraint on birth weight. You can't just have infinitely large babies because there's this skeletal rigid constraint on getting the baby out. How has this curve probably changed since 1951? We have bigger babies because when your baby is large enough that it's going to have a problem, what do? They just give you a C-section. So we've relaxed selection on this side. So we've removed that morphological constraint. What's happened on this end?
I feel like you have a fair number of premature babies that survive.
We do have a fair number of premature babies that survive now because we have neonatal intensive care units, we have little incubators that provide them a nice, warm, moist environment to live in. Our rate of getting premature babies to live is much better than it used to be. So premature babies are doing better, and large babies are surviving because of C-sections. And that's evolutionarily a problem. Not the premature baby one, but this one over here. Evolutionarily, why is that a problem?
Babies just keep getting bigger and bigger.
And what happens to the female pelvis? It stays the same because the constraint also shaped the evolution. These individuals are not surviving well because they are too big to fit through their mother's pelvis. If you remove that selection, these two things that would have been evolving in concert with one another, building a genetic covariance over time, now are decoupled. You can be a small woman, produce a giant baby, and both you and the baby survive. What happened in a lot of these cases where the baby perished? Mom probably perished also. And so females that had a large disconnect between baby size and pelvic size didn't get to reproduce after that. Those genes that govern that, which are probably a variety of different genes, then didn't go on into the next generation. So we're actually removing this morphological constraint that used to be embedded in human birth. So what we should expect to see is over time, we'll have more and more cesarean sections simply because we're beginning to decouple birth size, birth weight, and the geometry of the human pelvis. There are also physiological and energetic constraints. One of the most classic of these is the size number trade-off. If you look at the size of babies versus the fertility rate of individuals, the bigger you are, the fewer offspring you tend to have, because you essentially have a big box of energy available to you in terms of reproduction, and this box is actually available to you whether you are male or female, although we generally think of this in terms of females. But if you have a certain amount of energy, you can divide this energy up and say, well, I'm gonna spend 3/4 of my energy on growth and maintenance. And the other quarter of my energy I'm going to spend on reproduction. So that's how much you've allocated to the act of reproduction. Well, now you can take all of that energy and put all of that into one offspring that's very large, or you can divide that energy into 2 offspring. Each offspring is going to have to be half the size of the offspring than if you invested all of your energy in a single offspring. You can say, well, I want 4 offspring. Well, once again, that means that they're now going to be 1/4 of the size, et cetera, et cetera, et cetera. The more you divide your energy across offspring, the smaller those offspring get. as the size of an individual offspring goes up, the number of offspring that you can produce goes down. This is the size number trade-off. It's universal in biology because of this very real energetic constraint.
So is that to say that a smaller individual would only need, say, half of that energy to be growth and maintenance? Is that kind of what you're
No,
saying?
so you can also trade off growth and maintenance with reproduction. So in life history theory, for example, there are kind of two extremes in terms of reproduction. There is what we refer to as iteroparity. Iteroparity is when you reproduce over and over and over again, you have multiple bouts of reproduction, versus simulparity, where you basically grow and grow and grow and grow, and then you reproduce once, and after that bout of reproduction, you die. Because these two endpoints that you arrive at in evolution, you tend to find that these individuals are smaller in terms of total body size. They expend more energy in growth and maintenance, and they expend less energy in reproduction. And the reasons for this are manyfold. One of the reasons is that if you're going to survive to reproduce next year after having reproduced this year, you spend less energy in reproduction and more in growth and maintenance, because the goal is to live to the next year. You don't have to spend a ton of energy in reproduction this year if you know that next year you're going to be able to reproduce again. Simulparity, this is the grow and grow and grow and grow until you get to reproduction, and then you basically spend all of your energy in reproduction and die. When it comes time to reproduce, you basically spend all your energy in reproduction, which leaves you nothing left over for growth and maintenance. These things tend to be big organisms, and when they do reproduce, they reproduce tons and tons of offspring. An example of this would be the sequoias. These large trees that occur out in the Pacific Northwest, they grow and 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 grow. They live for hundreds and hundreds and hundreds of years, and then when they finally get to the point where they are ready to reproduce, they reproduce. There are pictures that you can find on the internet of a single sequoia tree putting out pollen, and there's a cloud there that's so dense that you can't even see the tree as it produces, and then after it does that, it dies. There are agave plants that are the same way, the century plants that supposedly grow for 100 years, and then they reproduce, and as soon as they are done reproducing, they die. As opposed to something like a mouse that in some cases might reproduce 2 or 3 times a year, and then will reproduce the next year as well. They produce smaller number of offspring. They're still investing quite a lot in reproduction, but they're always saving some energy back for growth and maintenance to get to the next reproductive bout. And so these are once again two ends of a continuum, but somewhere in there leaning towards the simulparous side of things, but not as far, are things like elephants that reproduce infrequently. They put quite a lot of energy into reproduction, and they produce offspring that are relatively large. They would be not on the iteroparous extreme of things. They would be somewhere in the middle, but moving on towards the simulparous side of things. Lower rates of reproduction in general, yeah.
So like a pig would be an iteroparous?
Yeah, pigs would be most things are actually. Insects typically.
Multiple litters of tiny little babies.
Multiple litters of tiny little babies is oftentimes what iteroparous individuals do. Humans are iteroparous, but once again, we aren't producing large litters of, so within this, a sequoia could produce one giant offspring. It doesn't, it produces gajillions of offspring. So within this scenario, you can have iteroparous species that produce relatively large offspring. When they do produce relatively large offspring, they produce single offspring. We are that way. Elephants are that way. We produce them less often. we invest more energy in them, and we produce relatively large offspring. So there are a set of decisions, and we talk about them as decisions, but the organisms aren't deciding these things. Over evolutionary time, evolution has shaped these organisms to reproduce in this particular way. And so there are a variety of different paths to reproduction. Different species have evolved down different paths depending on their ecology. If you live in highly variable environments, it might behoove you evolutionarily to reproduce multiple times a year, so that as you produce offspring, if something happens in a variable environment, such that your first litter of organisms all die because of a drought or something like that, later litters will survive. And so there's a whole thing in evolutionary biology looking at life history theory, looking at this difference between heteroparity and similarity. What are the ecological conditions that would favor heteroparity versus similarity, et cetera. So I don't know, did that shed any light on your question?
Yes.
Okay, all right, good. There are genetic constraints as well. The constraint that we saw in the snakes, where over evolutionary time, evolution, the selective environment, has shaped that population such that the only individuals that survive are individuals who are striped and crawl in a straight line, or checkered that reverse themselves often. That has set up this genetic correlation, this genetic covariance. Because it is a negative genetic covariance, that is by definition a genetic constraint. You can't have striped snakes striped snakes that reverse themselves a lot. You can't have checkered snakes that go in a straight line, because those individuals who do that tend to be weeded out of the population, so you end up with this genetic covariance, this genetic architecture, such that you tend to, when you find snakes out in the field, you tend to find striped snakes that go in a straight line, checkered snakes that reverse themselves. Questions about multivariate evolution? This stuff is unfortunately not in your book. This stuff is in the Stephen Arnold chapter for the most part, stuff that we've just been through. Now we're getting back into the book, where they talk about how you go about testing hypotheses in animal behavior. We've already talked about some of this. We can look at comparisons between individuals. Where have we talked about this in the most detail so far?
Shrikes.
Yeah, we've talked about this in the shrikes. We looked at the behavior of individual shrikes, looked at shrikes that were then experimented on, so we can do this through experimentation as well. But in the Shrike paper, it was experimentation that was looking at differences between individuals that were imposed upon the individuals by the researchers. But we can also do comparisons among species and see how one species does something versus how another species does something. And we can look and see, well, why does one species do one thing one way and the other species does that same thing in a different manner? So going back to the Joseph and Pinchot situation, this was what they did. They were looking at differences in the reproductive success of individual males that were placed under these experimental conditions. And so this is an example of definitely looking at individual behavior. But another way of doing this is actually to look at what's happening across different species. The example in your book is in the breeding behavior of gulls, and it looks at the black-headed gull, which is a ground nesting species, and the kittiwake, which is a species that nests on the cliffs. What are the differences in terms of getting your chicks to survive if you're a ground nesting bird versus a bird that nests on these sheer cliffs?
Need more camouflage on the ground.
Okay, so you might expect the ground nesters to have eggs that are more camouflaged, or perhaps chicks that are more camouflaged, because they're just laying out there in the open. And so that would be one contrast. What are some other contrasts that you might expect?
There's
more fear of predators or less fear?
Fewer.
Oh, fewer, okay, all right.
Sorry.
I missed the fewer. Yeah, so because they are on this sheer cliff, it's harder for predators to get to the nest, and so the parents don't have to spend as much time guarding the nest as opposed to a ground nester that might. The behavior that they have when a predator comes around might be very different. What other things might vary? Luke is looking in his book right now, so he knows all the answers. And if you're looking at the PowerPoint, you can also see the answers. So here we have two species that basically differ in terms of where they lay their eggs. And so the adult response to predators is they take flight early, they have alarm calls, they attack the predator. The kittiwakes remain on the nest until the predator is close. Rarely do they exhibit alarm calls. They do not defend their nest very much, probably because they don't have an evolutionary history of having to do that very much because of where they nest. Nest construction is loosely built, shallow cup. The kittiwake builds this deep cupped nest. Why would you do that on a cliff? So the chicks don't fall out. Not only that, these things typically lay eggs that are very pronounced rectangular, so that if you set them on a table and roll them, they will roll in a circle rather than rolling in a straight line. And this is really commonplace in cliff nesting birds and not as commonplace in ground nesting birds. When birds defecate, they oftentimes produce uric acid, which ends up being white after it dries. You've seen this on your car. As a result, black-headed gulls that nest on the ground, they don't go to the bathroom near their nest because all that does is call attention to the fact that there is a nest that is there, and the kittiwakes defecate elsewhere. The adults remove the eggshells after one of their offspring have hatched, because once again, the color inside the eggshell gives away the location of the nest. Adults don't remove eggshells. Cryptic coloration, not cryptic, weaker claws, strong claws, yada, 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 yada. Where you nest influences all of these aspects of the behavior, the morphology, egg morphology, cryptic nature of the eggs and the offspring, et cetera, et cetera. All of these things are affected by essentially where the species nests. Now, where you nest evolved in concert with behavior relative to defecation, behavior relative to removing eggshells, cryptic nature of the offspring, et cetera, but they're all related to the environment in which the species nests. And you can think about nesting on a cliff versus nesting on the open ground and see why these things would have adaptive value for those particular species. Now that's a simple comparison of comparing two species that do things like nesting behavior in very different ways, but we can also look at other things as well. When we look at primate ecology, there's a study that is mentioned in your textbook by Clutton, Brock, and Harvey, where they looked at both ecological and behavioral data. They considered a series of alternative hypotheses that might explain the behavior of different primates, but then they tried to look and see whether or not these ecological variables would be important in explaining the variation that they saw, or was there a phylogenetic signal such that the evolutionary history of the organisms might also provide some insight. And so they looked at things at a generic level to try and get at phylogenetically independent contrasts. So basically what you have here, this is a figure from your book. They essentially looked at things like body weight and testis weight. The larger you are, typically, the larger your testes are. But they have these things coded. The open circles are monogamous. The open triangles are single male systems. The cross is us. And the closed circles are multi-male breeding systems. When you look at this general correlation of male body size and testis size, Do you guys remember what residuals are in a regression? So something like this is going on. What are residuals in a regression?
Like the R-squared value, the, kind of how far they are away from that predicted line.
They're how far they are away from the predicted line. What it's basically saying is that at a given size, at this size, these individuals have much larger testes than these individuals do. These individuals have positive residual value. These individuals have negative residual value, and the further away from the line is, the larger the residual value is. So because body weight and testis weight are correlated with one another, different species are gonna differ in testis weight because they differ in body weight. But the question isn't that. The question is, at a given body weight, who has larger testes? And what do you notice about all of these solid dots, which are multi-male breeding systems?
Most of them have positive.
Most of them have positive residuals. In some cases, those positive residuals are really high, which means that for your body size, you have much larger testes than other primate species. When you look at the open triangles, these are single male systems. They have generally negative residuals, but they're all relatively close to the line. Open triangles, open circles are monogamous species. Monogamous species tend to be on the small side. Notice how the open circles only go up to here. They tend to be on the open or on the small size overall, and they tend to have 1, 2, 3, 4. 4 out of the 5 species in this graph are in the negative residual size. Why would mating system affect testis size?
Well, in a multi-male system, would there be more competition to reproduce?
Yes, if you have a multi-male mating system where females have the opportunity to mate with multiple males, how do you succeed as an individual in terms of reproductive output, the number of offspring that you sire if you are a male?
Continued reproduction?
Hmm?
Just continued reproduction to try and.
Continued reproduction, which means you need to be producing often, producing sperm often. Not only that, if you mate with a female and she has the opportunity to mate with another male shortly after you, or has mated with another male shortly before you, what happens among the different sperm from male 1 and sperm from you? They compete with one another, and so if you're capable of putting out a lot of sperm and the other male is not capable of putting out a lot of sperm, you just simply overcome that male's ability to successfully mate with that female simply because you flooded the market with your own sperm. So in multi-male systems, you should expect primates to have larger testes simply because of this concept called sperm competition, which we will talk about when we get to sexual selection later on in the semester. If you're monogamous, you don't have to produce a ton of sperm because you are in a monogamous relationship, and as a result, there's nobody in theory to be competing with. Now, as we see when we get to mating systems later in the semester, monogamy often isn't nearly as monogamous as we think about it being. In animal behavior, we oftentimes use the term serial monogamy, where you're monogamous for one breeding season, and then the next breeding season you're monogamous, but you're monogamous with a different partner, et cetera, et cetera. But even in situations where individuals are relatively monogamous, there is also opportunity for cheating. And one of the things that happens when you cheat is oftentimes you end up having higher reproductive success than when you don't cheat. And both males and females cheat in these systems, yeah.
So, in a multi-male setting, you know, one male mates with the female, and another one comes along. After that mating has occurred, those sperms compete.
Oh, yeah.
What about if the first male to get there, that breeding time is shut off? You know what I'm saying?
Oh, so that would be a situation, and we'll talk about that also. There are times when after male has mated with female, the female will no longer be receptive.
Yeah.
And so even if a male tries to mate with female, she won't want that, and she will try and dissuade him from mating. In some cases, they don't have any choice because their sexual dimorphism in the male is much bigger, et cetera, et cetera. Interesting situation in ducks. What do you guys know about duck mating?
I know they have corkscrew.
They have crazy reproductive tracts. They have all these curly blind alleys, and they mate with multiple males. And guess what they're capable of doing? These female ducks.
Choose which sperm.
They choose which sperm go down which channel. And so if you have you ever seen ducks mate?
Do they do it on the fly?
No, they do it on the
Oh.
ground.
Oh.
But what happens is the female runs away from the male eventually tracks her down, climbs up on top of her, bites her neck, and basically kind of forces her to copulate with them. If she doesn't want that male to fertilize her eggs, she just takes his sperm and directs it down a blind alley. And so she'll mate with multiple males, but because of her evolved reproductive tract, she can direct individual males into different parts of her reproductive system and only lets the sperm of the male that she wanted to mate with fertilize her eggs. Crazy, isn't it?
She has like locators on sperm and never.
No, she knows this male just copulated with me.
So like it went to slot A, slot B kind of thing.
Yep. So it used to be referred to as cryptic female choice, rather than choosing who you're going to copulate with, because you don't have any control over copulation. You then choose which sperm gets to fertilize your eggs, essentially. And it was cryptic because biologists didn't see the female choice going on, even though it's a system that has all the hallmarks of female choice. Colorful males, drab females, mating displays by the males, et cetera, et cetera. And so many of the things that you expect to see where female choice is a factor, but it didn't appear to biologists that females were actually making obvious choices. And it's because they weren't making them externally, they were making them internally. So this comparative method, looking across different genera, looking at the phylogeny of organisms to figure this out, has a series of limitations. Oftentimes, the people who do this don't pay very much attention to the alternative hypotheses. Everybody can be guilty of this, not just people who use the comparative method. Ecological variables are oftentimes not adequately quantified. Correlation does not necessarily mean causation, and these things don't typically consider that there might be multiple adaptive peaks or the presence of non-adaptive behavior. This is something that animal behaviorists have always struggled with. Some behavior is behavior that appears to not have any particular adaptive function. It doesn't necessarily function to increase the fitness of the individual. And then phylogenetically diverse species are not independent of one another. So if you are phylogenetically related, you might have a behavior that you got because you share a common ancestor, not because it has an adaptive function in its current situation. So if we look at, for example, mammals, these are all of the primates. These are all primates from here on up. And we are here. The other great apes, the chimpanzees, gorilla, the bongo. These are all in their own clade. These are the non-ape primates, old world primates, and then I believe these are all new world primates here. You can see that individuals that are phylogenetically related to one another might have a set of traits that are simply because they possess simply because their common ancestor possessed them, and they all got it through a common phylogenetic history, in the same way that there may be traits that all of the great apes possess because they have a common phylogenetic history. So you can do things a particular way. You can have a particular set of behaviors because of convergent evolution, phylogenetically different organisms, phylogenetically unrelated organisms have a particular set of behaviors because they happen to live in places that are ecologically similar, and so they're encountering similar selection pressures. Or you might have a trait because you simply inherited it from your ancestors, and so anybody who is phylogenetically related to you will have similar behaviors. People back in the 1980s started dealing with this in ways using what are called phylogenetically independent contrasts. If you have a phylogeny of a group of organisms, you can look at what the characteristics are that you are interested in, an X variable and a Y variable that would normally be correlated, but you can remove the phylogeny from this by basically looking at how does this species and this species differ from one another with reference to these two different characteristics. How do these two species differ from one another in terms of these two characteristics? And you don't plot the raw data of the species themselves, you plot what the difference is between those species. With phylogenetic methods, you can predict what the phenotypic state of the ancestors would be, so you can also do phylogenetic contrasts between these ancestors that even though they're no longer around because they ended up evolving into new species, you can still infer what their phenotype would have been. And then rather than having 1, 2, 3, 4, 5, 6 values that you're using to form this correlation, you have 3 values which are each of the contrasts. At that point, if you observe a correlation between whatever variable X is and whatever variable Y is, then that correlation isn't due to shared phylogenetic history because you removed the effect of phylogenetic history by doing these contrasts. So this is something that's sometimes difficult for students to understand. So what I did was I went onto the computer a couple of years ago and I ran some simulations. This is a program written by a guy named Liam Ravel, who is at UMass Boston. He has a program that allows you to generate a phylogeny of a set of theoretical organisms. So this is a phylogeny where we have 2 major clades that are separated by a long evolutionary history. They divided a long time ago, went off on their separate tracks, and both groups have recently undergone a kind of an adaptive radiation. Very rapid evolution after a long period of time of being separate. Along each of these tracks, along each of these evolutionary histories, you can program the computer to do what's called a random walk of phenotypic variation. A random walk just begins at a particular place, and then a random number generator determines where you go. Do you go up or down in terms of phenotypic value? You can do this for trait 1, and you can do this for trait 2, and tell the computer to basically do a random walk along this phylogeny. Basically saying, for this long period of time that organisms were on this point here, on this arm here, this branch here, what kind of phenotypic change would come if it was just kind of randomly moving around. Then when they divide, how much change occurs here, how much change occurs here, et cetera, et cetera. And so you independently evolve one trait along the path of this phylogeny, and then you independently evolve another trait along this phylogeny. Well, what you get when you do this random walk is you get a set of data that look like this. And if you run a regression line through that, you find that the X variable is negatively correlated with the Y variable, which would indicate that for whatever reason, these 2 morphological characteristics are negatively correlated with one another. What's a potential problem with this? Because we know that these are groups of species that have shared evolutionary history. Maybe these genetic correlation, or these correlations between these phenotypic traits aren't due to ecology driving the system. Maybe it's because if we map that phylogeny onto these data points, this is that big difference in the two clades. This is clade 1, this is clade 2. So if we looked at this, we would find that all of these species up here belong to that clade, and all of these species down here belong to this clade. So you don't have a bunch of points telling us that there's a negative correlation. You have one point that is all the individuals in one clade, and another point with all the individuals in the other clade. It isn't, and if you look at the points in this clade, it appears actually that if you just look at this clade by itself, it appears that variable X is positively correlated with variable Y, not negatively correlated. And so what you can do is you can run phylogenetically independent contrast, where you can say, how does this species differ from that species? That's contrast 1. How does this species differ from that species? That's contrast 2. How does this one differ? That's contrast 3, et cetera, et cetera. Do this for the entire phylogeny. You generate a ton of contrast because we have a ton of taxa that we're dealing with. Guess what happens to this correlation? It goes bye-bye because the original correlation was simply driven by these two different clades being different and being separated by a long evolutionary history. So this is actually a contrast for the X variable versus the contrast for the Y variable. And so all of the signal that you saw in that earlier correlation was just due to common phylogeny, common evolutionary history. Had nothing to do with anything because there is no correlation. So what ecological traits, what ecological circumstances would be driving no correlation? Well, obviously there's no correlation, so there's nothing being driven here. And so that's one of the advantages of using phylogenetically independent contrast is you can remove statistically the effect of this shared evolutionary history. And sometimes the correlations remain after you do this, and in some cases those correlations disappear. If the correlations disappear, the only thing that you were measuring is the phylogenetic signal in the data. If the correlations remain, you've controlled for phylogeny, so that correlation must have some sort of explanation. You can start paying attention to that correlation rather than just having what is essentially a correlation between two large clusters of points that are separated from one another, in this case in a negative way. So we can apply this to any particular group of organisms. We looked at the size number trade-off earlier. We can look at the size number trade-off with just the raw data, but we can also look at the phylogenetic independent contrasts on those same data. These are the phylogenetic independent contrasts that are from weaning size and fertility rate data. And after removing the effect of phylogeny, we still see that there's a negative correlation. The larger your offspring are, the fewer offspring you have. This then is now something that we can say, well, this is something that is real that we should pay attention to, because after we controlled for phylogeny, the correlation was still there.
So removing correlation that's due to phylogeny, and there's still a correlation that indicates that there's an ecological correlation?
Well, in this case, we know that there's an ecological cause for this, or a physiological cause, which is this allocation of energy. How do you allocate energy to offspring? Well, you can take the energy that you have for reproduction, you can devote it all to a single individual, or you can divide it up and devote it to a lot of individuals, but in the process of doing that, they're all gonna be much smaller. That's what leads to the size number trade-off. If this correlation disappeared after you controlled for the fact that these are all haplorhines, these are humans and these are the non-human apes, maybe this negative correlation is simply because you have a large cluster of haplorhines here and you have these groups down here. Same kind of thing, you have this negative correlation, part of which is explained by the fact that these are all phylogenetically related and these are all phylogenetically related. don't think of them as a bunch of individual species, think of them as 2 points, the haplorhines and the great apes. 2 points, easy to build a correlation between 2 points. So then, if you control for the fact that all the great apes are related to one another, and all these haplorhines are related to one another, after you control for that, if that correlation is still there, then you have to say, why is that correlation still there, even after we've removed the effect of common evolutionary history? Well, the reason is a good energetic reason that we went through earlier. It's not complicated, it's not hard to wrap your head around, but it's a real thing. It's not just an artifact of shared evolutionary history. There's a paper from, gosh, this would have been back in the 1990s, I guess. I'd have to look it up. You guys know about black widow spiders? The female eats the mate. As a result, what is the difference between the size of female black widows and male black widows? She's gigantic compared to the female. And so for the entire history of the study of arachnology, I'm done. This is the end of this presentation, so I'll just tell you a story and then we'll be done. Let's go look this up. This is a cool example of using phylogeny in things. So all throughout the history of arachnology, people have asked the question, you know, why is there this extreme sexual dimorphism? Why are these females so much bigger than the males? And the story that I was taught when I took arachnology is that the males are small because male dwarfism evolved over time to basically escape the clutches of the female. And so you started off with these big spiders, and over time the males got smaller and smaller and smaller as a way to escape predatory attempts of the female after they mate. And this is the story that was told to generations and generations of arachnologists, until Coddington and his colleagues came along and basically said, well, let's look at the phylogeny of spiders. For a lot of spider species, there's very little sexual dimorphism. Males and females are roughly the same size, but for some clades, there are big females and tiny males. And this is, if you blow this up, you'll see how these traits are distributed. Sexual dimorphism is distributed on this tree. And what they found is that when they looked at situations where there were large females and tiny males, in many cases, a given clade, all of the species in a particular evolutionary branch would have that characteristic, such that it might be a characteristic that was just inherited from a common ancestor. But by doing phylogenetically independent contrast, they could control for that. And what they found was that after controlling for that, there weren't many ecological factors that accounted for this, but because they were also going through and recreating the ancestral states to do these phylogenetically independent contrasts, they found that in almost all cases, and it wasn't uniformly this way, but in almost all cases, the common ancestor of groups that had this strong sexual dimorphism of giant females and tiny males, they all came from common ancestors where females were tiny and males were tiny. And so in whatever year this was published, it was the late 90s, they published this paper in Nature, I think it is, which is a big journal for a bunch of spider biologists to be published in, where they basically showed by referring to phylogeny that this was not a case of male dwarfism to escape predation, this was a case of female gigantism. Well, what are the advantages of being a gigantic female?
You can have more babies.
You can have a lot more babies. You have a lot more energy stores to put into reproduction. And in many of these cases, they divide that up into a lot of little babies, hundreds and hundreds of little babies. So what was the ecological driver of this sexual dimorphism? It wasn't predation being exerted by females against males. It was actually just selection acting on reproductive output on females, and that reproductive output selection didn't occur on males, and so they just stayed the size that they always were. Phylogeny is informative in figuring out why organisms the way they are. Giant female or dwarf male? Giant female was the conclusion. And all the archaeologists were like, huh, guess we're gonna have to change what we teach our students now. Questions? Yeah, look.
Did the females still try to prey on the males when it was that
Um,
common ancestor, when they're the same size?
Having studied spiders in the field quite a lot, spiders eat spiders a lot in the field. Male spiders eat female spiders eat male spiders, even in species that have not much sexual dimorphism, like the wolf spiders that I study. Oftentimes, you can see behaviorally when you see them mating, as soon as the deed is done, the male just runs like hell because it knows that if it sticks around too long, it's going to become dinner. Females, so female wolf spiders are these ground running spiders where they have an egg sac, they carry that egg sac around. When the eggs hatch into little baby spiderlings, the spiderlings crawl up onto the back of the female and they ride around on her abdomen for a couple of weeks afterwards, and then they slowly start dispersing. Well, over that time when the baby spiders are on the female's back, you can try and feed those female spiders crickets to keep their weight up, and they won't eat. And so a period of time after their babies have hatched, they undergo this period of appetite suppression where you can't feed them crickets, you can't feed them other conspecific spiders, you can't feed them spiders of another species. And then that lasts until a little bit after the babies have crawled off of the back. But if you wait like 3 weeks after that and take one of her own babies and put it in front of her, she will gobble it up like it's just some unknown spider. And so they go through appetite suppression, presumably to suppress their appetite so they don't consume their own young. And then after that passes, they'll consume even their own young because it's just a spider to them. It makes good sense for a female to eat a male after mating because then whatever energy and nutrients that male had, the female now has. So that's good for reproduction. But it behooves the male to not allow that to happen because if that happens, then he is now simiparous in a sense. He has gotten to reproduce once and only once. And so, cannibalism in spiders is well studied. As a matter of fact, one of the challenges that I had when I was in graduate school was raising spiders up from baby spiders because they tended to get to about the 3rd instar and then they just died from developmental anomalies. It's because they weren't getting a very well balanced diet. We figured out that if we could feed our crickets total cereal, it improved the health of the crickets so that when we fed them to the spiders, we went from like 10% survivorship to about 75% survivorship. When we talked to this group who was in a lab at the University of Cincinnati who had really good success at rearing babies, the way they did it was they just left all of their baby spiders in a common container until they got past that 3rd instar. And the reason that they survived so well is that they were just cannibalizing one another. One of the things about a spider is a spider has everything a spider needs nutritionally. And so they suffered a lot of mortality due to spiders eating other spiders in the lab. One of the problems with that was that then they were exerting selection on their population of spiders. Because they were looking at evolutionary things, they maybe shouldn't have done that. Because while they were getting high survivorship, the survivors were not a random selection of the babies that were born. It was the babies who survived the Hunger Games, so to speak, early in their development. Other questions? So spiders tend to cannibalize one another, regardless of sexual background. Other questions?
On the test,
Yeah.
the Arnold paper,
Oh, yeah.
they went into a little bit more of the weeds, if you will, of that.
They did. Yeah, and so they talked about maternal effects, which we didn't talk about at all, things like that. The main things that I want you to get is what's in the first part of that, the first 2/3 of that chapter,
Kind of the same stuff.
where it's laying the ground for how to think about multivariate evolution.
Okay.
We did mention genotypes by environment interactions, which are important, but we covered them relatively quickly. He talks at some length about them. Those things are important because oftentimes, as we talked about in class, the environment that you're reared in particular, might have behavioral effects that are quite profound. So I gave you the examples of the development of song and bird and how being denied hearing bird song early in development during this window of template formation, as ornithologists refer to it, that has catastrophic effects for the birds to be able to develop normal song when they get to be adults and things like that. They all have the same genes, they've just been denied the environment that helps song develop. Otromas. Yeah.
You think you can attach a key to that? I mean,
Well, that would be less fun. Why don't you write a key based on that? The only question,
well, mainly for the multiple choice.
the only question that is not covered in there is the winning question. Everything else that's in there, we've talked about.
Yeah.
So a good study opportunity, if you have a group of people that you would like to study with, like the non-Chiefs fans in the room, would be for you all to sit down and figure out what the key would be to that particular thing. You learn more from finding your own answers than you do from me giving you the answer. It's been shown through lots of educational research.
I mean, I was mainly talking about the multiple choice because it's a single answer, and if you're thinking that one's right and it's wrong.
It's a single answer, there's only one answer right, so it should be easier. You can't complain about multiple choice in your first year and then complain about multiple choice in your junior year.
I don't complain about it. I was, say the right thing here.
Write the key and come see me and see if you got it right.
Yeah, I guess that's
But
probably what I'm gonna do.
my guess is that your peers can also help you with that just